Webinar on neurotechnology. My name is James. I'm a neurosurgery resident in London, and I'm one of the Ideal webinar leads. We are delighted to have such a strong interest in neurotechnology, which really lies at the intersection between device innovation, first in human, and clinical translation. So the agenda that we have, it's going to be a busy hour session. So firstly, we've got the welcome and introduction, and then Dr. Arsenio Pérez will talk about the Ideal D framework for device innovation, and then closely followed by Dr. Esme Dola, who will give a talk on the Stentro device and human trials. We'll then close off with a 15-minute discussion and questions chaired by Mr. Hugo Layard Horsfall. And hopefully we'll finish promptly at 7 p.m. Just a few points of housekeeping. So this webinar will be recorded. Please stay muted if you're a participant. Of course, if you're asking a question, you can unmute, but save all questions to the question and answer session at the end. We really want to promote inclusive participation. So if you do have a question, please ask it. It's now my pleasure to introduce Mr. Hugo Layard Horsfall, who is a neurosurgery resident in London. Hugo is currently doing a PhD at the Francis Crick Institute and UCL, where he's a clinical doctoral fellow. Hugo's PhD seeks to record single neuron activity in the human brain. Hugo is also a founder of the Impact Network, which fosters cross-disciplinary collaboration in BCI research. Hugo, I'll hand it over to you.
Great, thanks, James. That's a very kind introduction, and well done for organising this. So whilst Arsenio is getting his slides up, I will introduce him. So Arsenio is a bit of a powerhouse in neurotechnology. We've been fortunate enough to have him in Ideal for a number of years, and we've worked quite closely together during that time. So he's a sleep neuroscientist and clinical scientist based in Montreal and specializes in EEG-based digital biomarkers, neurotechnology, and brain-computer interfaces, or BCI. He has a particular interest in neurodegenerative disease, and in relation to Ideal, he is the Ideal brain-computer interface project lead and a member of the Ideal collaboration leadership. He's also, in one of his other hats, the executive director of the Canadian Sleep Society. So I feel he is in a perfect position to start things off. So over to you, Arsenio. You might be muted.
Thank you very much. And that sounds far more exciting than I feel. But thank you for the great intro. And of course, even in 2026, we're still telling each other, unmute your mics. Yeah, so I'm here to tell you about Ideal D and why this is so important as we move from innovation to evidence in neurotech.
I think you've muted.
There we go. Yeah. Sorry. We're gonna look at how we move from innovation to evidence in neurotechnology with Ideal, and to kind of take a whistle-stop tour of where we are these days. And we have to really think about where neurotechnology is in 2026. And it's really kind of the same picture you would get when you open a fire hydrant and everything gushes out. There's so many advances in neurotech right now, whether it be implantable neurotechnology for persons with ALS or Parkinson's or epilepsy, for example, or non-implantable neurotechnology, for example, for people who suffer from depression or live with insomnia or have cognitive difficulties. We're also seeing that AI has really led to an incredible acceleration of innovation. Also an incredible acceleration of the commercialization of this innovation and of regulatory clearance of new devices. It seems like there's something being brought out to the market every day, or even sometimes twice a day. And this has also led to this rapid increase in consumer neurotechnology. But the landscape evidence isn't necessarily able to keep up with this. The innovation that we have is outpacing our traditional evidence frameworks. So we have to start really thinking a bit differently about how we approach this. And I'll give you an example. You may have, for example, someone who has ALS, and loses the ability to speak. And that person may receive an implantable BCI where the system now records their neural activity or their cortical activity, uses AI-driven algorithms to decode the language that they intend, converts those signals into synthesized speech in real time. The potential impact is enormous. They can restore communication for someone who's lost it, improve their autonomy, their quality of life. But how do we evaluate performance? It's complicated, right? Because it's going to depend on many things. The skill of the surgeon who implanted it, the learning curve of the surgeon, the stability of the signal over time, things that are very specific to the device, things that are very specific to the surgeon, how the AI model adapts over time, how things are calibrated, the how durable the devices are, how the body reacts to the devices. One of the devices, for example, that would implant some of the sensors in a stent, will that be rejected by the immune system? Software updates, and of course the human factor, how things get used in the real world, how humans react to it. It all becomes very, difficult to keep track of. So we might even look at other examples, things that don't involve implantation, something non-invasive. An older adult, for example, in the early stages of Alzheimer's, might receive a wearable EEG headband. This is, for example, something I work with all the time. That will stimulate the brain. That device may be detecting oscillations in the brain during sleep in real time. It may be delivering a certain kind of auditory stimulation during those oscillations to try and change how the brain works, to try and increase the clearance of neurotoxins to improve memory. The potential impact is great. It could improve that person's cognition. It could even slow their neurodegeneration. But again, how do we figure out and how do we evaluate the performance and the effectiveness? It's complicated. It depends on so many things. The precision of the timing of the stimulation, how it gets used in the real world setting. People may not use this device at home the way we intended them to use it, or we saw them use it in the lab. How the signal quality changes over time, how people adapt to it. Again, the human factor, the human component. So it gives us all these key challenges. The biggest of the key challenges, I think, we have these devices give us lots of change and lots of data. And it makes us really ask important questions. How do we evaluate these continuously evolving systems? And how do we do this responsibly and ethically across their life cycle? Where do we start? How do we know if they're ready to be evaluated in a certain way? And for which patients and for which outcomes? Which are the right outcomes? Will there be enough time? Some of these devices evolve so rapidly. Will there be enough time to gather evidence for them before the device is obsolete or changed so much that it's now a new device? And what about human factors? Will people trust the device? Will they trust the information they get from it? Will clinicians trust it? Will they be difficult to learn how to use? So of course you can say, well, that's easy. Do an RCT. We can do an RCT. That'll solve everything. Maybe we should. Maybe we shouldn't. Maybe not now. Maybe never. RCTs are not magic. The randomized control trial is not magic. It can't answer all questions. And we have lots of trials, but sometimes very little insight from them. What we need is better trials that fit better with neurotechnology. Because what we're going to find out, we're going to talk about, is that the key here with neurotechnology is to think about the specific technology, where it is at that moment of its development, who it's being used for, and for what outcome. So why we need a better way? Of course, we know the pharmaceutical model works. We've been doing that for ages. But surgery, the devices, and especially the neurotechnology, they're not static products. And I think that's the big difference. They're very complex. You already know as surgeons, the outcome of a surgery doesn't just depend on the surgery itself. It's also your skill with it, your experience with it, the patient you're working with, how you've had to adapt things, the setting. The devices have the same things, the characteristics of the device, how providers use it, how the algorithms adapt, how they're being used differently than they maybe were intended. How, all of these things are important. And also with neurotech, we have the problem that the devices are evolving continuously. Even if the physical device, implanted or not, hasn't changed, the software is changing continuously while it's being used. And it's giving us this overwhelming amount of real world physiological data throughout use. And that's something that has not typically been something we really think about when we think about evidence in healthcare interventions. So the result being that our traditional frameworks just can't capture this innovation or the real world adaptation in the way that we need them to now. So what I want to present to you is the ideal framework. And for people who already know the ideal framework, this will just be a quick refresher. For people who don't know the ideal framework well, I am so excited to tell you about it because I obviously am a big fan of the ideal framework. And what it gives you is this integrative pathway, a stepwise ladder. It's a more logical, sequenced way to organize innovation and organized evaluation. So whether you are developing your device or whether you're trying to evaluate the device or the neurotechnology, it gives you a nice operationalized sequential approach that helps you ask the right question at the right time and figure out the right approach to answer it. It really helps to fill this gap that we have now between these new ideas and interventions and this rapid evolution and how we actually gather technology for it or gather information for it. What it tells you, it asks you key questions, and at each stage is a very important question that it asks. And we're going to talk a little bit more of these, and I'll give you some examples of them. So for now, for example, we have this first stage, this preclinical stage, and what we're really asking is this neurotechnology ready for its first in human evaluation? Is it safe? This is where we're really doing all that preclinical testing, and we'll look at a good example afterwards to let us know, yes, we're ready to try this in a human being. Then we go on to the first stage. Can this work safely in human? We're going to prove the concept that this can actually work and is relatively safe in humans and may be worthy of further study before we move to this ideal two stage where we're now looking at whether this neurotechnology is stable or is the workflow stable? Do we know really what it's doing and in whom we should be using it with in a small setting, in a small number of participants or patients before we then expand this and try it with more participants or more patients in different settings? Now we're asking, is this reproducible? Can we use this across users, across centers, across environments? Then we'll go on to the ideal three, and that's again now that's more your traditional control trial, and we'll get to that a bit, where we want to see, is this better than what we currently have? Is it, how effective is this? And finally, and I think one of the most important things, especially for BCI right now, is this safe or effective in the long term? What happens when something, kind of sales, when something really scales into real world use? Not now just in our clinical trial, but when people are using it in the real world every day. So that first really important stage zero, that preclinical, this is an important stage, and this is where you often see things that are done with simulations, with animal studies, cadaveric studies, even sometimes health economic modeling. When you're trying to see if this device works, will it be feasible based on cost for it to ever be developed? And in neurotechnology in particular, we're really looking for safety, for usability, potential human factors. For example, things like the implant biocompatibility, the stability of electrodes, which has often been a kind of one of the sort of challenges in implantable neurotechnology. Things like wireless transmission, battery, hardware durability, signal integrity, that's also been an issue. Will there be issues with neural decoding performance over time? The synchronization accuracy. Will the AI algorithms be robust? Will the calibration actually work? So these are all things that you want to see, that you would want to test before the first in human. And an example of this, and this is again, this is a good example of something that would count as an ideal zero. This is a great one where someone's looking at simulating closed loop deep brain stimulation for Parkinson's disease. And specifically, for example, and this is what they're telling you, is that they want to know, will this work? Will this work before it's implanted to human? It's an in silico. What do we need to do to optimize the control parameters of this so that we know that it's actually likely to work and will likely work safely before it's tried for the first time in a human being? And what do they find in this? For example, in this one, when they test these algorithms with their in silico methods, what they find is, yeah, it's it actually does work. It actually does do what it's intended to do when in a simulation. So it's giving them the first step for driving the next step, which would be a clinically suitable closed loop system. So it's a way of showing what you would do before you try this for the first time in humans so that you have a good, reasonable chance of knowing that it's going to do what you intended to do in the first human use. That will then take you to that next important stage of ideal. Now it's ready for first in human use. You want to prove the concept. Before you said, okay, I've tested all this will work a certain way when I simulate it. What happens now in a human brain? Or in a human peripheral nervous system or wherever this is being used. And this is your proof of concept. Can it work safely in humans? Or sometimes can it work safely in a different population than it's been used before? Or if it's in a new variant of something that's been done before, will it work the same way? You're really looking for early feasibility. And it tends to be case studies, or a case study, very few patients. Really, it's setting up this idea and the system for systematic data collection and getting you to think about appropriate outcome measures that can go forward as this technology develops. It's really looking at early safety, early effects, and whether something is worth continuing. If something fails, that also is important because then you get to ask, why did it fail? You may have to go back a bit to figure out why it failed and to try and improve it. So even a failure is a success in some ways. An example that you might find a paper of, for example, is this real-time brain-computer interface for people walking with an exoskeleton. And it's a one-subject design. They had one person, they wanted to see how this would work in a human being. And they were looking for the performance, but also important thing, they were reporting that no adverse events occurred. So they already had the signal that it's feasible. It did work in this first in human trial, and there were no adverse effects. So it's actually something that's ready to be developed further with further research, maybe with more patients and eventually even in more settings. So this just gives you an example of things that you can apply and find when you look at that stage. And if things go well, of course, then this device and this technology can go on further. Now you've proven the concept, but now you start asking other questions. Is it stable enough? Is the workflow stable enough? Often we've got the device stable, but the workflow may not be stable. And are we really sure that that we know who it should be used for? Is it really ready for more patients? Are we sure that we don't need to do any more calibration, any more refinement to test its feasibility? Typically, what this becomes is more prospective studies. These might be single center case series where we're looking at patient selection criteria more closely, things that changed, things that we had to change, and why, really documenting that so that others learn faster and more safely, but also to prevent unsafe or devices from continuing on and even reaching the market because we don't have enough evidence for them. Another interesting example of this you can find sometimes is, for example, the command study where they're looking at an early implantable BCI for people with paralysis. And what they're looking at is, they had tried this before, they had a first in human, and now they say, okay, let's try this now with 5 people instead of one and see, is this going to work in these 5 people? Are there any unexpected events, anything we should know about? What they find is after in their study is that it did work as they intended it to, and that it's suitable for long-term neural signal acquisition, not just in the lab, but at home. So it's worthy of further study. They've tried it now with more people in a slightly different setting. It works. Now it's ready for further development, and they may be ready for the next stage. Another example using this with children, same thing. They want to see, is this ready? Are there any unexpected things we should be looking for? And what they find is, yeah, this actually works well enough. Continued exploration is justified. This is an important thing that Ideal brings to you, is this idea of making sure that before you go on and do this with more people and before you go on and do a trial, that it should be done, or that maybe that it should be paused and we should explore some of the issues. Because you also want to make sure that it's reproducible across users and centers and environments. Things often work really well in one center. It's when we explore them to different populations or different places that some of the issues arise. This is where we move into that next stage of Ideal, that 2B, where we're really looking at learning curves. Yes, the device worked beautifully with 5 participants. I used the device, I developed it, I can do it beautifully. But what happens when Hugo does it, or what happens with Esme does it, or what happens with John or Marsha or somebody else tries it? Are there learning curves? Are there differences in the performance and the quality? Are we using the right outcomes? Are we actually really, are our criteria for our patients correct? Maybe we're sometimes using the wrong patients. Are there any things that patients can tell us? For example, you might have a great device and patients say, I'm not using that. you know, Maybe a wonderful device, but we have to understand how patients will react to it. Um, this is an important stage because it's also helping you think about a future control trial and think about issues that you may run into to make and help you make a better trial in the future. And these again are these collaborative prospective cohorts now where you're really looking at more providers, more patients, more settings. So a good example for you, this was just published in JAMA, I think in 2025. And what you have here is this multi-center perspective study where they're really implementing this adaptive deep brain stimulation for Parkinson's. And one of the major finding is it was tolerable in a range of participants in a range of settings. It was effective and it was safe. They looked at the stabilization of their algorithms, their learning curve. They optimized. They looked at how this would work in the real world. They built consensus around what the actual best outcomes were, the best biomarkers, and it really helped them transition toward the next stage where they compare this stimulation to something else. So this is a great example of how Ideal can help you really move into that next stage. The next stage we all know well, the randomized control trial, and there are so many flavors of it. It's like going to Baskin Robbins. There are 34 flavors of trials for you. But so we don't really, Ideal's perspective is really, you want to know if this technology is better than what's currently there, and you want to look at effectiveness and cost effectiveness as well. Usually those are RCTs compared with some alternative or placebo. There's lots of great guidance already in the world for clinical trials. What Ideal tells you is use that guidance. There's important guidance already. Consort and others, use that, but also add other things. Focus on fidelity, for example. How well did this go? Did the neurotechnology not work well because people weren't using it as they intended, or the settings were changed beyond what was intending, or is it just that it didn't work? What was actually done? Document everything. What was the equipoise? You may do this study and find that patients do not want to be randomized. If you say, I want to randomize you to this, dual orexin receptor antagonist for a pharmaceutical, for example, for your insomnia, or the stimulation device, people might say, I don't want the device, give me the pills. And some people may say, I'm not touching pills, give me the device. They may not want to be randomized. So it's an important thing, or you may have centers that won't do it. So this is really an important part that I want to take you to. It's such an important part, and it's something we don't have enough of yet, partially because so much of the neurotechnology is so new. But one thing that Ideal emphasizes, you don't have to wait till the device has already been out for 10 years to do this. This can start from the very beginning. This is one of the things that I think is important about Ideal. Start thinking about collecting this data, building the registry for this data, building the thinking about collecting this data early on as the device develops, so that when you get to this longer term, you're now looking at long-term safety and effects in the real world. I always love the work that I've done in the laboratory and the work I've done in the real world, because they are neither here nor there sometimes, and so we really need to see how things work in the real world, the durability, indication creep, how often we see medical devices moved beyond their original purpose. We're going to see this a lot with BCI, especially in the consumer BCI. We need to start thinking about that and things that are unforeseen. Late effects, rare adverse effects we wouldn't have seen until 20,000 people were using the device. Differences in their outcomes, different people respond differently. We can look at subgroups, really looking at problems in delivery over time. And this could be by observational studies, registries, sometimes even our trials with a very long-term follow-up, and incorporating things like mixed methods and qualitative work to understand people's experience. A good example is the product surveillance registry. This was just published in Neurology, developed specifically to look prospectively at device performance and outcome data in deep brain stimulation. And this is a really great example of how this is being set up, but we need to draw more attention to it. So I want to leave you with some final takeaways about this. Neurotechnology, it's evolving so rapidly, it's increasingly complex. I want to pitch you that Ideal, especially the Ideal D, can help neurotech innovation, can help give us a safer runway, clearer milestones, stronger, more transparent evidence that includes more patient input and protection without slowing innovation down. But we want to hear from you. We need your input. There are lots of important next steps, expanding Ideal to these brain-computer interfaces. But tell us your opinions. We want to hear it. We want to have this conversation, and I highly encourage you to join it. And keep in mind our annual conference in November in Oxford, because we would love to hear from you. And thank you for listening.
Perfect. Thanks, Arsenio. There's lots of content that would be useful and exciting to dig into, but mindful of time, and I'd like to leave some time at the end for more of this discussion. But it's a beautiful overview of some of the issues facing BCI at the moment. Super relevant. So without further ado, and your talk leads us nicely on to Esme, because you mentioned the COMMAND study, which is the synchron stentrode study. So Esme, I've known for a while. She studied in Cambridge and then did some clinical training in Oxford before seeing the light, or not, depending on how you look at it, and moved across both stateside and industry, and is now Associate Medical Director at Synchron, which, as you all know, is endovascular brain-computer interface. They've been pioneering on some of the world's first longer-term endovascular implantations, and moving on towards neuromodulation, preclinical development, and clinical trial design. So Esme, the floor is yours. We're very excited to hear your talk.
Thanks so much, Hugo. What a kind introduction. Let me get my slides up. Probably could have done that before. Oh, nope. Those are the wrong ones. Okay. Is that showing for you guys? It's showing for me, but
Not yet. No, it's great. Thanks, Esme.
okay. All right. So thanks everyone for joining. So I'm Esme, as you said, I'm the Associate Medical Director at Synchron. I trained in the UK, did lots of research in the UK, and then decided to jump to industry. Dark side, not dark side. Yeah, whatever everyone's views is on that. But it's been great. So very excited to talk about how Synchron sits in the broader BCI landscape or implantable BCI landscape. You know, what we've done so far, where we're headed, long way to go. These are my disclosures. So I think, you know, most people already know this, but just the level set and start with the basics. So I'm specifically talking about implantable BCIs today, not considering non-invasive modalities at the moment. So these are any neuroprostheses that interface with the brain to restore lost motor and or sensory capabilities for people with paralysis. This is motor BCI. So that involves placement of electrodes on the brain, which has traditionally been done via craniotomy. And that has produced pretty remarkable results. So, you know, the BrainGate academic collaboration in the US has really driven forward some incredible progress, as well as now many other groups across the US, the UK, and the Netherlands, and China should be mentioned. So, you know, some groups have achieved computer cursor control. So what this would involve is patients undergoing motor intent. For instance, they might imagine, you know, pronation of their right arm, which is then decoded into leftward movement of a cursor, and supination, which is decoded into rightward movement of a computer cursor. Those other groups have really expanded into the control of the physical domain, which we do know is quite important, especially for patients with spinal cord injuries. So there's lots of work on robotic arm control. There's an academic citation here, but should probably congratulate our competitors who are doing some really incredible work as well in their Convoy study. So Neuralink is doing some great efforts on robotic arm control. So a lot of efforts working on somatosensory stimulation. So lots of groups trying to restore somatosensory sensation, which has already been shown to improve functional upper limb control. And I think some other really exciting work has been speech decoding. So one of the, I think, seminal papers by Nicholas Card has been a use of the BrainGate 2 system, which is a microelectrode array based BCI, which penetrates into the brain parenchyma. That sits over speech cortex. And while patients are doing imagined and attempted speech, this managed to decode at a pretty good pace, so 31 words per minute, the intended speech with 97.5% accuracy and a really impressive vocabulary. There's been other, you know, incredible efforts in speech decoding in the field, especially by groups at Stanford. And there's been, you know, some industry work on this as well, spinning out from groups using an ECOG-based approach. But the speech decoding has really seen pretty remarkable leaps. So challenges would you then, and you know, that's why I think the ideal frame was very interesting. You know, we have all of this academic proof of concept, and it's been really remarkable. You know, we've demonstrated someone can fly a drone using just their thoughts. You know, as a headline in the Daily Mail, it's pretty impressive, right? But how do you then translate that into something that's meaningful for patients, that has regulatory acceptance, that gets reimbursement? Yeah, that's a big leap. And so there's many implantable BCI companies that are all in early stage clinical trials. So us at Synchron, but also Neuralink, Precision Neuroscience, BlackRock, who've been around for a while, Paradromics, Axoft, Neurosoft, InBrain. There's lots of really, you know, there's a ton of excitement and appeal right now. And a lot of these are still in early stage, but trying to get to pivotal. So these companies generally take one of three approaches. So the most traditional approach uses penetrating arrays, which are inserted directly into brain parenchyma via craniotomy. So these give fairly high resolution signals. You can get action potentials, but obviously it's more invasive. So several companies do have trials underway, but none of these have published clinical results yet. The other thing I think to flag on approaches with craniotomy is that it might be a limitation for patients who can't undergo general anesthesia. So some more progressed ALS patients would fall into this category. And obviously that is a really early use case or clinical use case for BCI. The other approach uses ECOG arrays, which are placed on the brain surface. So this is still via craniotomy, but it can be a much smaller incision. It's been called a microslit approach. The idea or the hope is that this would be more stable over time because you're recording local field potentials rather than individual action potentials. And in theory, you would have less neuronal death. But current trials are still limited to temporary placement. So there's no take-home BCI independent use yet. And then our approach is endovascular. So we deliver the stentrode through the jugular, track it all the way up to the superior sagittal sinus. We sit right next to the precentral gyrus. And the idea is that we record signals through the vessel wall from motor cortex while patients are intending or attempting movement. So the idea is that we're leveraging a pretty well-established clinical pathway. There's cath labs all over the place now. There's in theory more cath labs than there are operating theaters, and there's in theory, there are more physicians that can be trained to deliver devices interventionally than neurosurgically. So we've done several early feasibility trials. I'm just going to walk you through these. So our very first study was a switch trial in Australia, which has already been referenced. Our company was founded in Australia. So that is where a lot of the really early translation happened. So there were 4 patients with severe bilateral upper limb paralysis, either due to ALS or PLS, who were implanted. The primary endpoint for that was safety, and all safety endpoints were met. And we noticed no serious adverse events and no negative impacts on lifestyle and some early signs of improvement in quality of life. But this is, that's not necessarily the right study yet for these kind of efficacy endpoints. So we then expanded to the US where we ran the COMMAND trial. So there we implanted 6 patients with paralysis from ALS or stroke. And again, this is still a safety study and all safety endpoints were met. Full 12-month follow-up is complete, but extended follow-up is ongoing. So across all these 4+, actually, I should say, sorry, the switch trial would probably translate to 2A in the ideal framework. I think it's a good example of it. Please correct me if I'm wrong, though. I'm in no way an expert on the ideal framework, but that sounds about right where that fits. And then, we're trying to push through as we go to our next trial. So very briefly across these 10 subjects, we had 0 device-related serious adverse event resulting in death or permanent increased disability. 1 subject did pass away from ALS regression, which was deemed unrelated to the study. And if we're including our secondary endpoints as well, we did 3-month CT imaging, which showed preserved vasopatency and no device migration. So 0 thrombosis, 0 stenosis, and 0 migration beyond 2 millimeters. Why do we care so much about migration? I just wanted to show some early preprint, early research that's out in a preprint on our procedural approach and our targeting. So as I mentioned, we target it neurointerventionally. This is all done in a cath lab. We have found when analyzing placement across 10 participants that there's a reasonable amount of variability in how it overlaps. So we try and aim the centrode to overlap as much as possible with M1 and then have the most distal part of the centroid overlap slightly with SMA. So you can see here the SMA, M1, and S1 have been mapped in these colors, and you can see the placement of the stentrode next to those. So from that and from some data I'll show in a moment, we've really identified that precise M1 targeting has to be a deliberate procedural goal. So it cannot be left up to chance because we know it's very important. Now, in that same work, we measured the stentrode to cortex distance. So some work done by Hunter Schoen. And we found it's about an average of 6 millimeters, minimums 3.7 millimeters. There's a reasonable range of 3 to 6 millimeters there. And that variability is really driven by individual vascular anatomy and also the degree of atrophy that these patients have. So the distance between the stentrode and the actual cortical surface is reasonably variable, but we can consistently get meaningful proximity. Now, why do these two things matter? Well, we looked at various factors, anatomical and clinical, to try and see what is the best predictor of signal strength. So both absolute signal strength and signal to noise ratio. And it came up very clearly that the degree of overlap with M1, with primary motor cortex, is by far the best predictor of signal strength. So that beat the distance to a cortical surface that I showed earlier, residual muscle strength, years since diagnosis, functional activity in S1, SMA, and various other clinical parameters. So the bottom line here is that the degree of M1 overlap is by far the most easily controlled variable that determines signal quality. And so we have various methods to try and ensure maximal precise targeting of our placement. And you can see here on the left, so here this is showing a burst count of a recording from our patient in a relatively high gamma frequency, so looking at 100, 200 hertz. And you can see that we're decoding these individual movement attempts into a single switch there. Right. So what can we actually do with that? So we've been exploring various key activities of daily living, and we know that, in today's world, the ability to control digital device is pretty integral to having digital autonomy and to your quality of life, especially for patients who are paralyzed, who spend much more time behind a screen than most of us. So the big thing that's missing here is there's no consistent outcome measure that can be used to robustly measure improvement on this, because there's just no such thing. No one has tried to measure improvement in digital autonomy before. It's a huge amount of effort in the field on this at the moment. A collaborative effort between industry, between academic partners and regulators to try and settle on an outcome measure that we can all agree on is clinically meaningful and improves patients' daily life. But we have demonstrated various key activities of daily living that patients are able to use digitally. So patients have used our device to submit a pain report on MyCharts, on online medical management systems. Environmental control. So patients have used our system to control, lights, AC, opening doors, and so on, which can be really critical for patients who have spousal caregivers instead of professional caregivers and might spend time alone during the day. Patients have used our device to do shopping. Communication has been a really key focus. So sending a message, for patients who are non-verbal, some even very basic communication becomes absolutely critical. So we have we've had lots of patients who during the day might use alternative assistive technology and might be absolutely fine with that. But then overnight, when they are in pain or in discomfort, or maybe they have a ventilator and they need suctioning, they have very fragile ways of notifying a caregiver. So some patients might grind their teeth, or other patients might, increase their thoracic pressure to make their ventilator alarm. And ALS patients are remarkably inventive, or they will figure out a situation that works for them some of the time. But what we're trying to do is give patients access to that ability all of the time, because that reliability is just really critical. Right. So just to flag our upcoming study in the US, which is called Intent. Here, we will be enrolling 10 patients with ALS. We're still looking at safety and early device feasibility. We've upgraded our hardware. So we're, I guess we sit in stage 2B of the ideal framework there. So exploration, we're broadening to multiple sites and a larger cohort. And we're really trying to learn about the outcome measures that might be useful in this setting as well. All right. And I'll leave this link up if anyone's interested in more info on our upcoming trial specifically. So thank you for your time. I do have to scoot off in 6 minutes, but if there's questions in the meantime, I'd be very, very happy to answer. Thanks.
Perfect. Thanks, Esme. Really interesting stuff. And there's lots to pick apart. There's the technical aspects of synchron and centroid. But I think whilst you're here, we've spoken a little bit about where synchron sits on ideal framework and bringing Arsenio in. Obviously, a lot of the device innovation happens in the States. And so these companies are following FDA approval or guidance. And when you refer to pivotal trial, you could also be talking about an ideal 2A, 2B study. So there's lots of similarities between some of the languages that we're talking about, we're using. What do you think are some of the barriers for some of the BCI companies to adopt something like ideal, which is already quite an established framework for surgical innovation? It's got quite a long track record. So yeah, first to Esme, and then Arsenio can probably come in on some of your thoughts.
So I guess maybe rather than being active barriers, it's probably that there's already existing regulatory frameworks and language that that companies use, which could completely coexist with this, right? But it's maybe about what people are comfortable with. So I don't know how many people are deeply familiar with the ideal framework yet. So it's an awareness thing. And then there's just like, language that's commonly used. So the FDA has an early feasibility study program. So people talk about going from EFS to pivotal. It's language that people are comfortable using. Maybe the other aspect is that, and this is my very uninformed view, but I imagine that the ideal framework is something that's really, really relevant for a select group of people that are taking a device from ideation all the way to clinical translation and to post-market use. And actually, most people that work at these companies have a specific niche that they're involved with, and they might only be involved with, half of stage 2B. So they might not necessarily have that wider bird's eye view, and maybe they don't need to. I think this is really relevant more for, I think it's really relevant more for those that are starting idea and trying to take it all the way. But that's that might be a very uninformed view.
And I would add to that. I think for one of the things I wish I would sort of give people the idea of that it is not a matter of slowing innovation at all. It doesn't slow it. It's part of it. But I think one thing that is also important is that I think what Ideal does, because it's sequential, it doesn't mean that you have to go from one to the other rigidly, but that I think that different people in different aspects of the life cycle of the device can find value from different components. So for example, someone may only be working in that 2A part. They may never be involved with what comes next, but it's having the information to think it ahead of potential problems that will arise even in that 2A part before they arise. So I think one thing that I would say about the value added is just it's sort of the thinking ahead component. We've just had the proof of concept. We've just done the bench testing. We're going to do our first in human. What should we be thinking of in advance to build into that first in human to have a more successful first of human? Or what things should we be looking at in advance? Because one of the things I think often happens is you have these early trials, first in humans or bench testing, issues will come up, and then you have to go backwards and think, oh, well, we didn't think of that, or oh, we didn't include an outcome measure for this, oh, we didn't plan to collect that. I think what Ideal does is it gets you to think about it in advance. You build it in early. So you have that when the data is being collected, which is a very different prospect than trying to do it in reverse. But I do agree. I think sometimes the issue is also that these things exist in a certain language in the EU or the FDA. I think that's part of it. I think what Ideal does is it enriches whatever stage you're in. So I think there's a lot more room for synergy there.
Great, thanks. Hani?
Yeah, thanks. And Esme, I thought it was a fantastic talk. I was really really keen to hear it, and it was lovely to see how that's always progressed. I'm conscious you have to leave, you said, like in a few minutes. If you don't have time to answer this, please don't feel committed to stay. But my question was actually just about the practicalities around accommodating for the learning curve when you introduce these implants. So, one of the ideas behind the ideal framework is to account for the complexity, a big part of that being the learning curve, both in in the technique, but also in refining which cases you select and which patients benefit most from the procedure. That's particularly true for BCI. So have you noticed over the clinical cohorts that you've already used this in, that that's iterated, that there have been changes in which particular patients you're picking off, or the particular techniques that are being used to introduce these?
Makes sense. So I guess you're asking both from a procedural side and then from more of a medical patient selection side. Yeah.
Exactly.
No, so
Yes.
absolutely. So procedurally, because we're there's no one else in the space, so we don't have anyone else we can learn from. We know there's venous sinus stenting as a precedent. So we've had great help from the innovators in that field, but otherwise we're sort of on our own. So the first few implanting physicians, especially in Australia, that implanted a device, and then now we have a few PIs in the US who've implanted several. We are massively learning from them and trying to set up, scalable training pathways such that we can take their learnings and apply those much more generally, because we're the only source of learning that there is. There's not really anything else to compare. So absolutely, we think that's critical, and really when it comes to scaling, making sure that procedural training is standardized and easily applied is really important. And I fully agree. And I think to your comment, Arsenio, I think one of the problems that BCI field is having is around outcome measures. And that problem might have not been there if we'd been taking the ideal framework in mind, 10 years ago, if the field had been considering that. That was just a deviation. On the rest of your question, Honey, in terms of patient selection, we're finding the same thing. We do know that, selecting patients who are engaged and motivated and who are, interested in science is really critical. And even in the academic literature, probably the best patient who's demonstrated clinical at-home use for a sustained period of time has been a patient by the Utrecht team in the Netherlands. And that's really what a lot of the foundational clinical BCI literature is based on. And she was someone who, she was a former physician. She was excited to do this for the sake of the science. We know that in these early feasibility trials, selecting patients like that is so important. And obviously we're refining our, more broadly as we go forward, we're refining our inclusion exclusion criteria together with FDA to make sure we're capturing an appropriate population.
Thank you.
Great.
That's really helpful.
Thanks. Prof. McCulloch?
Hi, Esme. I was really interested in the two studies you've done and what you learned from them, because of course, in early studies like this, you have to emphasize safety and feasibility. But what you're actually interested in is how can we make this better? How can we optimize it? And so I'd like to learn more about, it seemed to me from your data, the things you wanted to do were to place this as close as possible to the M1 area and in the right space, 3 to 6 millimeters from the cortex. So did you learn anything in these 10 cases about how to do that better, faster, more easily? That's the sort of information that I think is really, really valuable from 2A studies.
Yeah, absolutely. So the answer is yes. So when it comes to precise procedural targeting, we've developed a really, really robust rigid co-registration protocol. I'm unable to comment too much on the details of that because this is a recorded talk. But the approaches that we have to try and make sure that we are within that 2 millimeter range that's been probably our biggest learning. Well, I can't say our biggest learning. There's been so many learnings. That's been a very key learning procedurally, and we've developed a protocol which we'll now be testing out in our upcoming trials on procedural targeting. Mm-hmm. Other things we've really learned a lot from patients. I'm not just saying that. I think there's a real risk of developing technology that looks cool and that's exciting, but that patients are not going to use. Already we know in the assistive technology field, loads of people use their eye gaze as a bookend. Some people it's massively meaningful. Other people use it as a bookend. So how do we make sure that when there's technology that has a lot of potential, that we're actually directing that to be meaningful to patients? And so that's been one of the biggest things that we've learned, and we've changed our hardware quite considerably to try and reduce any burden for caregivers in terms of setup, in terms of involvement. So that's been the other learning. If patients are going to use it, it has to be simple, it has to be easy, it has to be straightforward and low barrier. And that maybe that should have been obvious before, but when you apply that to specific technology and a new patient population, learning exactly what is annoying and what isn't annoying is really critical. I think our clinical learnings have been pretty big there, and they're ongoing, right? You know, we're still learning a lot on how to guide our product development to actually serve patient needs. So we're designing with them and not for them.
Another important benefit from the stages two is that you get that, because I think that's the same thing with the non-implantable ones that I work with. They work beautifully in theory in the lab, but when we have older adults, for example, try and use them at home, some of the issues that arose, we couldn't have expected. So, and in retrospect, even in those trials, we thought, I wish we had gotten more of outcomes, built that in first, tested it a bit first before, so you don't have 300 people emailing you saying, the device keeps slipping off, and it turns out they weren't strapping it on the right way or something that, or it's uncomfortable when I sweat. These things that you don't know, and I think you get so much more out of from the 2As and 2Bs before you go on to larger trials.
Absolutely. And that's exactly the stage that we're sitting in now. I'm so sorry, I have to drop off. But thanks so much for having me. It was absolutely lovely.
Thanks, Esme. Great. Lovely to see you. Good luck.
Thanks. Bye.
Great. So I think I can see Parsons got their hand up, so I'll come to Pass in a moment. But just Arsenio, thinking a bit more about adoption of Ideal. So you mentioned that it's been used for complex interventions like surgery, and I would argue that some aspects of surgery are less complex interventions than BCI, and we still struggle to adopt Ideal into more regular workflow when it's perhaps just surgeons and a few engineers. When you've got big VC money coming into small trials, quite centralized in geographical location and in certain healthcare systems, how do you think we can really encourage people to understand more about Ideal and to apply it to neurotechnology and specifically implantable BCI?
Yeah, and I think what we need there really is first we need to show it. So I think it's something that we're working on, in trying to kind of partner more with people who are developing these, so that we can have an ideal informed trial, for example, or an ideal informed example. I think the big issue really is that with these BCIs, because they're evolving during the trial, they're evolving while they're being used. Even if the hardware hasn't changed, even if the implantation hasn't changed, the software is, and it's responding constantly to biosignals that are constantly changing. So I think that that presents such an issue that our way of gathering the evidence has to change continuously, because the device and the software is changing continuously. So I think part of the challenge more is getting the broader community to understand this is something that Ideal is uniquely adapted to, because we're not using a rigid framework that's built on a static product. I'm not saying the pharmaceuticals are 100% static, but by the time you put a pharmaceutical out for trial, it's static. It's relatively static. These are changing so often. So I think what we need to do more, or would be helpful, is also to get people to really present this idea that this is a fluid framework built specifically to encapsulate that constant evolution and change. And we've seen that with the way that we deal with AI and surgical robotics, for example, this particular way of thinking. So I think it's partially sort of let's work together with BCI more and with Neurotech more to show how this works in reality, because I think it has specifically has a really good contribution to make that I think is unique to Ideal.
Yeah, agree. Thanks. Very, very nice answer to a challenging question, I think. So we have a hand up from Parsa. I hope I'm saying your name correctly.
Yeah, hi. So I really wanted to ask a question from Esme, Dr. Dole, but I guess there wasn't. If anyone else would be able to help me out here. So we know with devices like the implantable BCIs with Neuralink, the intracortical arrays degrade over time due to neuroinflammation. How does that work with if you're stenting it, and if you is there any data from Synchron on needing to how are you going to manage the signals getting weaker and weaker over time? Is there data on 2+ years? We know Neuralink did a study recently about the arrays and the signal getting weaker. Is the same thing going to be happening for Synchron?
So I think for Synchron, the aim of the game is to actually have endothelialization of the stent into the sinus. So any patient who has a stentrode is on anticoagulants for a certain period of time, at least dual platelets or dual agents, and then they come down. Some of them are on these antiplatelet agents anyway. But the aim of the game is the endothelialization, so you expect there to be some integration of the stent into the blood vessel. How that affects signal quality, I haven't seen any data. I can't comment on it. But I think it would be amiss to suggest that there would be no degradation over time. And just for BCIs, particularly implantable BCIs in general, is not just the actual recording site of the electrode, it's all of the other electronics that go along being in contact with the human body. So one thing that Neuralink has, which is quite strong, is that it's a wireless device with no battery. It's just literally the implant which sits on the surface of the skull and intracortically. Stentrade, for example, like some of the other devices, they have a wire like DBS, which then requires a pocket, so therefore you have an infection risk there. So there's lots of other surgical considerations other than just recording site and recording device. And I think this is something that Synchron get asked all the time about the antiplatelets and surely this increases risk of stroke, etc. And so I think there's no one right approach. They all have different philosophies, they all have different pros and cons. And obviously I don't work for any of the companies, just as a disclosure on this. I don't know if, yeah, sorry,
Yeah, I can add for Synchron because it was something I was also very interested in with, and they have I don't think they've published it yet. They just have it as a preprint, but their 5-year follow-up data, what they say is that they have had that they've maintained signal integrity, that they haven't had much degradation. That they kept signal stability for the motor imagery classification in home for patients using it at home. They also have, I think they did publish their 12-month data and showed that they had remarkably stable signal stability. But I think those are the kind of issues that, again, from an ideal perspective, you build that question in early on, not just in the ideal zero when you're doing the simulation, but you start building it as you go. Because these things, these life cycles are incredibly rapid. A device now may be gone in 3 years. It may be superseded, but even within that period, you're still building up this, collecting this data, starting this registry, looking at that through every stage. So it's not just that you get to 2 years later and say, oh, now we start collecting prospectively to see signal stability. No, we can be looking at it from the very beginning. By that point, you've already had 2 years. So I think it's one of the important contributions also.
So I just mindful of time. I know James said we had to finish, but just on that point, Arsenio, I think it's a really good point, and it's something that I grapple with on a regular basis when trying to bring in ideal. So essentially, if we had a crystal ball and could understand all of the outcomes that are important, for stage 1, stage 2, stage 3, stage 4, it would be perfect and actually be great for the rest of the world. So I just wonder how you resolve that in trying to understand and build in early what these outcomes are in the long term, because it's important. But for such novel technology, actually, it's very hard to predict, even with the best multidisciplinary team in the world. You might not get it all. So how do we understand this and keep the innovation going?
Actually, I know Peter has his hand up, and I don't know if I'm gonna let Peter. I'll add something if there's time, but I want to let Peter have his.
Okay, I'll be brief, Hugo. I mean, you're absolutely right. I think that if I was starting again with Ideal, I would introduce two things as requirements in stage 2A. One would be a systematic risk analysis and risk mitigation strategy for whatever it is you're doing. I mean, this device, obviously, I'm thinking as an upper GI surgeon who knows nothing about neurosurgery, isn't it a little dangerous to leave something floating there in the sagittal sinus? I've heard of sagittal sinus thrombosis. It's a bad thing, you know. And whether there's a system you could work out how dangerous this is pre-implantation, pre-stage 1, stage 2A. And the other thing which Esme I think was actually emphasizing is human factors.
Yes.
I would, if I was rewriting 2A today, I would say you got to get a human factors analysis of how this is going to go, and including, of course, a patient's perspective. And those two suggestions, I think, would give you some of the things I feel you're trying to get at, the crystal ball,
Yeah.
So
And I
that's
couldn't
my
agree more.
contribution.
That's where I was going to go also, that it's having this human factors, but also having this other stakeholder perspective, because we can also learn a lot from other things. For example, it may not be the same implantable device, but some of the effects and some of the patient responses and difficulties you may have with a neuroimplanted device are maybe similar to something that you would have had with an implantable device elsewhere in the body. And we know this absolutely for things like the non-implantable BCI. They've been out for a while. I always want to remind people BCI is not new. In some ways, this goes back to the 1960s. So we have lots of experience. What we have to do sometimes is also bring some of that experience in early on. So there's no reason, for example, that even at a stage 0, you couldn't have a potential participant, patient, human factors voice in there, because for for example, I won't say with whom, but one of the companies was developing their own device. I work with them on sort of this aspect, and one of the things I say to them, "This is lovely. This is fantastic. Will the little old lady who's 81 years old with mild dementia be able to put it on, or will you be able to keep her from taking it off? Because it's a wonderful device, but it looks a bit bulky. Oh, we didn't think of that. Well, let's ask one." So
Yeah.
sometimes you can think of these things in advance by thinking of the end user, which is, I think, not something we think of. We usually think more of, let's get the device together, let's do this, let's do that. Oh, when we're doing the trial now, we'll get end user feedback. We need that early on, because even if it hasn't existed yet, you will already save a lot of problems by thinking forward early. And I think so. I think we can do this. I think it's just it's a more of a failure of imagination sometimes, but we can do it.
Perfect. Great. I think on that note, James, did you have anything to say at close?
Yes, I've just got the link for feedback. So thank you everyone for attending. A special thanks goes to the speakers, of course. This QR code will take you to the feedback link. Once the feedback is filled out, we'll be able to issue everyone with a certificate of attendance. We'll also be sending the feedback link out via email. I also wanted to signpost 2 future events. So we have another webinar coming up in September on robotics. And we also have a face-to-face conference that will be happening in November. It's actually November the 9th and 10th in Trinity College, Oxford. So please do attend if you can make it. And yeah, that's it.
Great. Thanks, everyone.
Thanks, everyone. Bye.
Bye, everyone.
Bye.